All right, so we talked about enzymes being catalysts. And actually, I told you that when we talked about enzymes, think two words. What were those two words? Protein and catalyst. They're made of proteins. Their function is to be catalysts, which means they help things get started. They help chemical reactions get started. They speed up those chemical reactions. They do that by lowering the amount of energy needed to start up the reaction. And that is called activation energy. Because they're proteins, their structure, very, very important. Structure determines function. Their shape has to be exact. If that shape changes, they don't work. That shape's held together in a lot of ways, just like in any proteins. Hydrogen bonds, because amino acids are polar, they can form hydrogen bonds. Disulfide bonds form between sulfur-containing amino acids. And also the hydrophilic interactions between amino acids. Some amino acids don't want to be close to water, so they fold away from water. Some do want to be around water, so they fold toward it. Those three things are the three main things that hold that shape into place. A key word to know when you're talking about enzymes is the word substrate. A substrate is something that an enzyme acts on. For example, an enzyme we talked a lot about last class, amylase. What is the substrate for amylase? Amylose, which is another name for starch. Where in your body does starch first come into contact with amylase? In your mouth, in your saliva. And amylase will take starch and eventually break it up into glucose. Starch is a polysaccharide. Glucose is a monosaccharide. Catalase is another enzyme we talked about a little bit last class, and it's one that the lab that we're going to do is about. Catalase is the enzyme. What's the substrate? Nope. Hydrogen peroxide. Okay? Catalase will take hydrogen peroxide and break it up. And it'll break it up into what? Two things. Water and oxygen gas. So all those bubbles you see when you put peroxide on the cut, those are oxygen bubbles. Another key word to know about when you're talking about enzymes is active site. The active site is a specific place on an enzyme that the substrate binds to. And they have to fit together like a lock and a key. It's not only the shape of that active site that's important, but it's also the charge. So active sites, shape and charge have to match up. Let's look at a picture of what we're talking about here. So here we see the enzyme. And I've asked you this question before. What would each of these little blue dots represent in this picture? If this whole thing is the enzyme, what is each blue dot? Speak up. Okay, not a protein, each blue dot. Amino acids, right? Because proteins are made out of amino acids. But this little spot right here, that's the active site. Notice it's got a specific shape, and that the substrate fits into that shape. We talked about that fancy phrase, complementarity leads to a high degree of what? Specificity. The idea that only one thing fits into this shape. And that because of that, each enzyme can only catalyze what? One reaction. It only makes one thing happen. Another key thing to realize is that once it fits in, the enzyme will slightly change its shape so that it'll work better. And that change in shape is a good thing. It's called an induced fit. Or sometimes it might be called what else? Yep, a conformational change. And that will actually help the enzyme work better when it changes its shape once the enzyme and the substrate get together. So let's back up a little bit. All right, we looked at this equation and how that, I put it in symbols I said E for enzyme plus S for substrate. They react together. When they react together, we call that the ES complex. When they're together like this right here, that's when the enzyme's doing its thing. It's working. Then they separate. And when they separate, the enzyme is unchanged. It can be used again. And what we end up with are products. So the substrate gets changed, the enzyme doesn't. It's important to know, most people think, when they hear the word enzyme, they think what? Breakdown. They think degradation. But I want you to make sure you realize that there's just as many enzymes that are synthesis as there are enzymes that are degrading. There are just as many enzymes that put things together as there are that take things apart. Make sure you know that. All right, now we talked about how enzymes lower activation energy, and we said they could do that in 3 different ways. One of those is they line up the substrate so that they bump the right way, they react the right way. Before any two chemicals can react, what do they have to do first? They have to collide. And normally they're just colliding by random, right? Heat. That's why heating things up make them react faster, because they're more likely to collide. But they not only have to collide, they have to be lined up the right way when they collide. If they're not lined up the right way, they don't react. So an enzyme will grab them and line them up so they'll react the right way. Another thing, enzymes can cause physical stress on a bind. Once the substrate fits in there and that conformational change happens, it sort of puts some torque on the binds. And enough torque, enough stress on a bind, what will happen to it? It'll break. And then the other thing is that it can change the substrate's reactivity. Enzymes can add things to a substrate. For example, what do kinases add to a substrate? What do kinases add to a substrate? They add phosphate from ATP, right? And by adding phosphate, what else do they add to that substrate? Energy. Other enzymes add electrons or take away electrons. What's the fancy word for adding in electrons to things? No, that'd be phosphorylation would be adding a phosphate. Adding electrons is called reduction. Oh, that's bad writing. Board's a little off up there. Reduction. R-O-L-U-C. Yeah, it's reduction. Just take my word for it. Reduction is adding electrons and also adding what else? What always comes with the electrons? Energy. Okay? Why is it called reduction? We're adding stuff to something. What gets reduced? The charge goes down. The oxidation number might go from like +2 to +1. It gets an electron added to it. All right, and then also some enzymes can take away electrons. Whenever you hear reduction, you should also think the other word. What's the other word? Oxidation. Oxidation is taking away electrons, taking away energy. Those two always go together. That's why sometimes we call those kinds of reactions, what kind of reactions? Redox. We talk about redox reactions. Because if one thing gets reduced, what had to happen to something else? What had to have happened to something else? It had to have been oxidized, right? Because electrons just don't come out of thin air. If you add electrons to something, you had to have taken them from something else. All right, let's go a little further here. I think we covered this whole thing. Complementarity leads to a high degree of specificity. Enzyme and substrate have to fit together. If they don't fit, enzyme doesn't work. The reaction doesn't happen. Each enzyme is only good for one reaction. And also, most enzymes are found in only certain parts of your body. That's because what can be turned on and off in different parts of your body? Genes. Genes code for proteins, and enzymes are proteins. All right, this is sort of where we left off the other day. The other day we were talking about cofactors and coenzymes. And we said that cofactors are minerals. What are minerals? They're not vitamins. They're usually metal ions.
Oh,
They're usually metal ions. They're metal ions that certain enzymes have to have attached to them so that they'll work. Zinc, iron, copper, calcium. There's a bunch of them in your body, and without those metal ions, certain enzymes don't work. That's why mineral deficiencies are bad. Coenzymes are basically the same thing, except coenzymes are these big organic molecules, not metals. We call coenzymes vitamins. They're large carbon-based molecules, but they do basically the same thing. They attach to certain enzymes, and without those coenzymes, the enzymes don't work. That's why vitamin deficiencies like scurvy, night blindness, and all those things occur, because without specific vitamins, certain enzymes just don't work, and you have all kinds of major issues. I want to go forward a little bit here and talk about these graphs. Here we've got a graph of rate of reaction graphed against temperature. And what we're seeing over here is that as we increase temperature, what's happening to the rate of the reaction? It's going up, right? It's getting steeper, steeper, steeper. Why is it getting faster over there? Heat makes things move faster, right? And because they move faster, they're more likely to collide and react. Then we get up here. It tops out. What do we call that temperature that it works best at? Optimum or optimal, either one. Sometimes both words are used. Optimum temperature. And in your body, in Celsius, what is that temperature for most of your enzymes? Well, in Celsius, it's 37. In Fahrenheit, it's about 98.6, right? Now realize that 98.6 is not the same for everybody. There's a range there. Not everybody's exactly 98.6. You're close, but not exactly. After we overshoot that optimum temperature, the rate starts to go down. What's happening at that point? And we call that denaturation. Our enzymes start to fall apart when they get too high. Those hydrogen bonds start to break. Shape starts to change, and it doesn't work anymore. The same kind of thing happens for pH. Most enzymes in your body have a certain pH that they work best at, an optimum pH, and we can see that here. For most enzymes in your body, what is that optimum pH? Yeah, it's about 7.35, which is the pH of your blood and your body fluids. Now there's some exceptions, like pepsin over here. Its optimum pH is somewhere around 2. Where's pepsin probably found? It's in the stomach, where it's really, really acidic. But most of your enzymes, the optimum temperature is about 7.35. And if you go too acidic, or if you go too basic, what starts to happen to those enzymes? Denaturation. They start to fall apart. So temperature or pH, either one can cause your enzymes to fall apart. And tomorrow, or Wednesday, we're going to bring in some big chunks of liver, chicken liver. We're going to grind it up in a blender, and then we're going to pour stuff in there. See what happens. It's kind of the most gross demonstration you'll probably ever see in this class, but it's good. It'll show temperature. It'll show pH. It smells bad. You ever fished with chicken liver before?
Yeah.
It stinks. Okay? It's not a pretty picture. We'll also do the same thing with potatoes. It won't be so gross. All right, I think we mentioned inhibitors just a little bit last class. That word inhibit should clue you into what we're talking about. Inhibit just means to what? Stop. Stop, prevent. Inhibitors are things that stop enzymes from working or prevent them from working. And we're going to talk about two different kinds. We're going to talk about competitive and non-competitive inhibitors. Let's start off by talking about competitive inhibitors. They're called competitive because they compete with what? For what? They're shaped like the substrate. And that means they can bind to the active site. So a competitive inhibitor is something that's shaped a lot like a substrate. It'll fit into that active site and block it so that what can't get there? The substrate. And if the substrate can't get there, what does that mean about the reaction? It doesn't happen. So let me say that again. Competitive inhibitors are substances that are shaped a lot like a substrate. They'll fit into the active site and block it. And there are a lot of them out there. Let's talk about one example that you may have actually used in your body before. There are these medications out there called sulfa drugs. Sometimes people that are allergic to antibiotics don't give these to when they get a bacterial infection. I hadn't told you what it blocks yet, but here's what these things do. Folic acid is a chemical that we need. It's also a chemical that bacteria need. What is folic acid? If you watch enough TV, you know this because it's on commercials all the time. You got to watch more TV. You got to quit DVR and things. You got to watch the commercials. Okay? Folic acid is a vitamin. It's a vitamin. It's a coenzyme. Certain enzymes in your body have to have it. Particularly, what are they commercial folic acid about? It's all commercials, orange juice commercials usually is where they talk about it. Orange juice has a lot of folic acid, and they talk about how it's really important for who to get it. Older people. Pregnant women. Okay? And it turns out that apparently folic acid is a coenzyme involved in making the nervous system, and the enzymes that make the nervous system. And if you don't get enough folic acid as a mother, as a pregnant woman, your baby might be born with a condition called anencephaly. Now let's break that word into pieces for a minute. What's an mean? No, or not. And this root you probably should recognize. What does that mean? C-E-P-H-A-L. Nope. Try again. Head. That word actually means without a head. Now, these babies aren't born without a head. But they're born, really just as bad, they're born without a cerebrum.
What? That's fine.
Now, the cerebrum is the major part of the brain. When you see a picture of the brain, the big part, that's the cerebrum. That's the part where all conscious thought happens. It's where all memories are stored. It's where all reasoning, it's where all emotion is at. It's the part of the brain that makes you human. These babies are born, they're alive. They're breathing.
I didn't know how to breathe.
Because that part is controlled by a different part of your brain.
Wow.
You have a part of your brain called the primitive brain or brainstem. That makes you breathe, it makes you your heart beat. But these babies will never be conscious because this is the part of the brain that causes you to be conscious. So they're born, they live sometimes for months, but they can never wake up. They can never be human. They can never feel anything. They're just like vegetables.
Yeah. So would they seek out like food and stuff?
Would they seek out food? No. They have no, they're not going to move consciously because they don't have any consciousness.
But they can get food stuff.
They're going to put nutrition into their body, into the blood. That's maybe the worst birth defect you could be born with. Yeah, I mean, sometimes they don't. But there's this line between abortion and not abortion. This is kind of where, what do you do? I mean, I'm as against abortion as any person on earth is against abortion. But in this case, what do you do? These people can't ever wake up. They don't have the part of the brain that makes them wake up. It's just not going to happen. So I don't know what you do here.
So is there no way to see this before the baby's born?
They probably could, but it's something that they probably wouldn't be looking for. But in an ultrasound, I would imagine they could probably tell. Not, don't quote me on that, but it's not something they would normally be looking for. What were you saying?
You laid it in on.
Oh yeah, just look up anencephaly and you'll be able to find it.
I'm saying.
Not now, because I'm in the middle of this recording thing with the PowerPoint.
That'd be cool though.
It would be cool, but it's gonna mess up my whole setup if I do that though.
Yeah, you can't like record and go on YouTube.
Alright.
Stuff
Yeah,
here.
not right now, no. Yeah.
So they have a head.
Yeah, they have a head, just the main part of the brain's not in there.
So it's gonna get it in.
So how is their head shaped? Is it like?
No, I know. Again, I haven't seen a picture of this in a long time. I think the head is shaped normally. It's open, hollow. All right, but back to where I was going. This folic acid thing. Bacteria need it. You need it. We usually get our folic acid from eating. It's a nutrient. It's a vitamin. Bacteria, on the other hand, have enzymes that make it. They make their own. They don't get it from their diet. They make it. Sulfa drugs block the enzymes that make folic acid in bacteria. Well, if bacteria can't make it, they have to have it. So what happens? If they can't make this vitamin, they die. They have to have it to live. These drugs don't hurt us though, because guess what? We don't have the enzymes to make it. Because how do we get it? From our diet. So these drugs inhibit enzymes that bacteria have that we don't. Therefore, it hurts the bacteria. Doesn't bother us. We don't have those enzymes. Does that make sense? It's a competitive inhibitor, though. These actually bind to the active sites of the enzymes in bacteria that make folic acid. All right, and again, the answer to this question, why doesn't this affect us? We don't have those enzymes. We don't make folic acid. We get it from our diet. Let's also talk about non-competitive inhibitors for a minute. They're not competitive, which means they don't bind where?
Like the active?
They don't bind to the active site. They bind somewhere else. But the net effect is the same, which means what? When they bind to that enzyme, what happens? Nothing. They turn it off. They block it from working. They just don't stick to the substrate. They stick somewhere else. Maybe the most famous non-competitive inhibitor is penicillin. We know that penicillin is an antibiotic. We know it stops bacteria from reproducing. But what you may not know, does anybody know what kind of enzymes in bacteria it inhibits? Most antibiotics actually do this. Most antibiotics inhibit enzymes related to making cell walls. Hey, we got them already. So those enzymes to make cell walls don't work, which means bacteria can't what? They can't reproduce. How long does an individual bacteria cell live? A few hours, maybe days. So very quickly, what happens to the bacteria? They die out. Why doesn't this bother us? Why does taking penicillin not make you sick? You don't have cell walls. Therefore, you don't have enzymes to make them, right? So we're inhibiting enzymes that bacteria have that we don't. And therefore, we, in essence, kill them off. Most antibiotics affect the bacteria's ability to make cell walls by inhibiting the enzymes that make the walls. All right, a lot of poisons are also enzyme inhibitors. Heavy metals. And we're not talking about Van Halen here, right? What are we talking about here with heavy metals? We're talking about lead. We're talking about mercury. We're talking about cadmium. Those are the three main ones. Lead, mercury, and cadmium. So that's Pb and Hg. What's cadmium? Cd. Those things are toxic, very toxic. And they're toxic because that once they get into your body, they bind to enzymes in your body. All over your body, but the famous ones are the ones in your brain, right? And they inhibit enzymes in all parts of your body, but specifically in your brain from working. And ultimately, if you get enough of them, they cause death. Arsenic also inhibits enzymes. Cyanide inhibits enzymes. the little toxin from those tree frogs, what do we call them? What's the name for those bright colored tree frogs? Say again? Dart frogs, poison dart frogs. Poison from those things are enzyme inhibitors. Even like chemical weapons, they work by inhibiting certain enzymes in your body and ultimately lead to death. One other thing to know about these non-competitive inhibitors, they don't bind to the active site, but they may bind to some other place. Like sometimes they'll bind to the spot where the cofactor or the coenzyme is supposed to be. Or they may bind somewhere else. But the key thing about non-competitive inhibitors, they don't bind to the active site, but the net effect is the same. When they're there, the enzyme doesn't work. It's inhibited. All right, last thing we'll mention today is this thing called a metabolic pathway. Big important word, metabolic pathway. Most things in your body take a lot of steps. Like cell respiration is not a one-step thing. Making of a muscle is not a one-step thing. It takes a lot of steps. Let me go to a picture here for a second. This is a metabolic pathway right here, this picture. We've got our first reactant. What do you think E1 means? The first enzyme. That first enzyme will grab onto that first reactant, and notice it's turning substrate A into something else, B. And then enzyme 2 will take B and work on it, turn it into C. 3 will take C, turn it into D, and so on until we get, at the end, what we're actually trying to make. This is the metabolic pathway. All the steps involved in doing something in your body. And typically, each step takes a different enzyme. And notice here we've got at least 5 enzymes involved in this metabolic pathway. Another important term to know related to these pathways is a term called an intermediate. An intermediate is the product you get from each step except which step? Except the last one. What's intermediate mean? Inter means between the beginning and what? The end. It's products you get somewhere between the beginning and the end. It's not what you're actually trying to make, but it's a step in the process of getting there. So like B here, that would be an intermediate. C would be an intermediate. D would be an intermediate. E would be an intermediate. But F rather is what? It's the end product. It's the thing you're trying to make. Which in cell respiration, the end product would be what? ATP. Okay. Let me back up a slide. Yeah, question.
Why are they both binding to the same enzyme?
Both binding to the same enzyme. Okay, this is another thing. Give me a minute and I'll get to that. Turns out that the product here is actually binding to that first enzyme and turning it off. It's like an on-off switch. This is not an active side over here. This is called a feedback side. And give me a minute and I'll get to that. All right, so again, a metabolic pathway is a series of steps involved in some kind of chemical process. The products that you get in between the beginning and the end are called intermediate or metabolic intermediates. And it's important to know that a different enzyme makes each step happen. Something that scientists have found is that a lot of these metabolic pathways are pretty much exactly the same in all known organisms. For example, cell respiration happens basically the same way in you as that it happens in plants, in bacteria, in fungi, and everything else. Protists also. Yeah, scientists would say that because those pathways are the same, that must mean what? Common ancestor kind of thing, evolution, right? Phylogenetics. A couple other terms we'll mention here about metabolic pathways. Some metabolic pathways are catabolic. That word catabolic means what? Break down, degrade. And other pathways are anabolic, which we know means build up. Right. All right, I'll mention this and then I'll stop for today. Feedback inhibition. This is real important. Does it make sense for all of your chemical processes to be happening full speed all the time? No. You got to have a way to turn them on and off. And that's what this does. This helps to turn on and off stuff. It's using negative feedback. Remember, negative feedback is when your body gets to where it needs to be and then it tells your body to what? Stop. So in this case, here's a good definition of feedback inhibition. Now it sounds really complicated. Read that definition. Just read it to yourself. Temporary deactivation of an enzyme brought about by an elevation of the end product of the pathway. So let's say that there's some pathway in your body where you're making something. Let's just say you're making glucose. And you got enough glucose. What should happen to the process of making glucose if you have enough? Stop it. Doesn't make sense to keep on. That's what this is. Make something until you get enough, and then turn it off. Save your resources. Don't waste them. Don't keep making stuff you don't need. This works a lot like a thermostat. It's a negative feedback thing. Make it, make it, make it, make it, make it. And then when you get to where you need to be, stop making it. Turn it back on later if you need to. Now the way this works, well, here we go. The way this works is that here's an enzyme. Here's a metabolic pathway. This pathway is making some product. Let's say that you've made a lot of this stuff. Let's say that each one of my little marks here represents one of these. So these are all our end products. Eventually these things just start building up everywhere. They're all over the place. The concentration of them just keeps getting higher and higher and higher and higher. They're everywhere. Eventually, one of these end products bumps into our enzyme. Usually the first enzyme in our pathway. And it bumps into a specific site on that enzyme. Not the active site, but a different spot. It's called the allosteric, or sometimes feedback site. And when it bumps in there and it binds to that site, what does that cause to happen to that enzyme? It causes it to change shape. And when it changes shape, what did we just do to that enzyme? We deactivated it. We turned it off. Notice that usually the enzyme that gets turned off is the first one in the pathway. Because if this one gets turned off, what does that mean about all the others? They're off too. Because this one only works if A got turned into what? B. And it didn't, because that enzyme's off. So this process of feedback inhibition works when the product of the pathway binds to a specific site, the allosteric site, on the first enzyme in the pathway. When it does, it changes the shape, turns that enzyme off. But is this a permanent thing? No. When the concentration of these products starts going down, what happens to the one that's attached up to this enzyme? It lets go, turns loose. And when it turns loose, what happens to that enzyme? It starts back up, starts rolling again, starts making more end product. So this is a good way for you to save your resources, conserve your energy, be green, sort of. This is like being green today. All right, let me turn this recorder off and then I'll get some questions. Sure. Give me a second. Let me turn this off.